Hello, everyone, and welcome to our webinar today. I'm Stan, CEO of MyLabs, and back 2015, I was one of the initial developers of our MiXCR platform. Today, Dmitry Chudakov, our scientific co-founder, Professor Chudakov, is joining as a panelist and will be happy to answer your questions at the end of our webinar. The topic of today's webinar is related to B-cell biology, and our speaker is a scientist, Artem Mikhailov, who works in this field for quite several years and collaborates with us and contributes to the MiXCR development by providing a constant and rigorous feedback from the scientific fields. So before we start, I wanted to say that the structure of the webinar will be the following. First, our speaker will give a talk and show some slides covering both technical aspects on how to run the software to conduct bioinformatic analysis and highlight interesting scientific results. After that, we will have a Q&A session. So during his presentation, please text your questions in the Q&A section of your Zoom window. So you can check, there is a button at the bottom of your Zoom window. You can push the button, and please do not hesitate to ask any questions which you find relevant to our today's topics. And at the end of the talk, we will answer all your questions. Also, if during the Q&A session you will want to ask your question using your voice, please push the raise hand button at the bottom of your Zoom window, and we will unmute you to ask question in person. So finally, we are making a recording, and every attendee will receive a link with the recording, with the slides, and with the links to the materials which we used which our speaker used to prepare this presentation. So now I'm muting myself, and Artem, please, your turn.
Hi, everyone. Thank you for joining us today on this webinar, which will be dedicated to the new features in MiXCR, which are mostly related to the B-cell receptor repertoire analysis. And I am Artem Mikhailov, and as Stas mentioned, I have quite a vast experience in B-cell receptor repertoire profiling. I've been working in this field for 6.5 years, and I also have vast experience in wet lab biologies, basically in sequencing libraries, kit preparation, and protocol optimization. And I've been helping to develop MiXCR for the last 5 years, mostly from a user viewpoint, as a scientist, providing the feedback and developing the inputs for the algorithms. And in a month, I'll be a postdoc in Stanford at Scott Boyd's lab. And during all of these years, I've been fascinated with the B cells, and I study the B cell receptor repertoires mostly. And during all of these years, I've used extensively the MiXCR software. So I'm extremely eager to present the newest features which has been developed and introduced in the newest version. And so most of you probably have heard of MiXCR or used it, and you may know that MiXCR is the most powerful platform for immune repertoire analysis in existence by now, and it offers a great flexibility. You can run it for various scenarios and various data sets, from RNA-seq data to targeted repertoire sequencing, or even now for the almost all of the single cell sequencing platforms. And with the recent versions, the unique molecular identifiers and cell barcodes and also sample barcode processing was introduced, along with the post-analysis and plotting modules. And most importantly for us today, the new features for B-cell receptor repertoire analysis were introduced, such as allelic variance, inference, and somatic hypermutation trees reconstruction. Also, what I personally like about MiXCR very much, that it only requires Java to run, so there is no need to hustle with any dependencies, no need to running any environments and so on. So you just download it and you just run it. So extremely handy. And today I will demonstrate the full pipeline of analysis with MiXCR, starting from the raw data from raw FASTQ files to the upstream processing and the clonotype tables to the downstream applications and biological insights. And I will be doing this using real-world data, a set of immune repertoires from a middle-aged, generally healthy donor, from which we obtained several time points during the course of several years. And what's peculiar, during the last 2 years, this donor had 2 COVID-19 infections with different strains, with Alpha and Omicron strains. And we have time points before the first infection, then 15 days after the first COVID-19 infection. Then we have time points before the second COVID-19 infection, and one of these time points is just right before the infection, 3 days before the infection, which was kind of lucky moment here. And then we have a time point 7 days after the second COVID-19 infection. So this is a peak of response called very nicely in this time point. And finally, one more time point 5 days after. So it's total 12 days after the second COVID-19 infection diagnosis. And most of these samples were collected in replicates, and peripheral blood mononuclear cells were isolated from this, and RNA was extracted to prepare sequencing libraries using my laboratory's human immunoglobulin multiplex kit. And below you see the structure of the libraries from this kit, and basically it covers almost the full length of the receptor, including, of course, the junction, the CDR3 junction, and also it allows to determine the exact isotype, including subisotypes such as A1 or A2 or subisotypes of IgG, and utilizes unique molecular identifiers, which will be helpful for us for correcting errors from sequencing and PCR errors. So these libraries were sequenced using Illumina MiSeq machine with 250 base pair paired reading. And today I will show how to process that kind of data with MiXCR, and then to get a meaningful biological insights from it. And to start with, I'd like to show you the running of upstream analysis with the MiXCR, and it is extremely easy these days because it requires just one line of command, which is MiXCR analyze, which takes the first argument as a preset name, and in preset name you specify basically the name of the kit with which your library was prepared. And now there are presets for all of the commercially available kits for immune repertoire sequencing, including the single cell platforms such as 10x or BD Rhapsody. And you can make also your own preset for your own custom protocol, which would account for all peculiarities of your library preparation. Then MiXCR analyze takes file names of the FASTQ files from sequencing machine, and I'd like to emphasize here that these are the raw files, so no pre-processing is needed, no UMI extraction or barcode extraction. Everything is now built in MiXCR. And finally, it takes an output prefix as the last argument, and this output prefix will be used in output file names of the files which this command outputs. And it actually outputs several files, most important of which is the CLNS file, and this is a memory efficient and computationally efficient binary file which contains all of the information about clonotypes which were found in these samples, in this sample. And this information can be easily then be exported into human readable format using MiXCR export clones command. And it also, MiXCR analyze also outputs the report, which contains all of the details and also the statistics about running the whole pipeline and each step and all of the issues which were with the data. And finally, the command outputs the clonotype tables in tabular format separated by chain. So here are several examples. So this looks easy, but under the hood, there is a little bit more complicated story. Actually, MiXCR analyze runs several separate MiXCR commands. And in this case, in case of amplicon data with unique molecular identifiers, these steps are the following: MiXCR align, MiXCR refine tags and sort, MiXCR assemble, and MiXCR export clones. And we'll go deeper about all of these commands. But I just would like to mention that for different kit and different pipeline, for example, for any kind of single cell processing techniques, for example, these steps might be a little bit different. So this is the pipeline tailored exactly for amplicon data with unique molecular identifiers. And first command which MiXCR analyze run is MiXCR align. It basically aligns all of the sequencing reads to a reference VDJ and C gene segment library for T and B cell receptors. And as I already mentioned, the multiple species are supported now in MiXCR. So there are gene segment libraries for human, mice, rats, and also non-model species. However, user can supply his or her own custom library for any species, and MiXCR can run this library as well. So also at this step, MiXCR extracts unique molecular identifiers and cell barcodes and also sample barcodes sequences from the reads, trims adapters and trims primers. So everything is performed by MiXCR, and to do so, a special regex-like pattern syntax was developed, and here you can see the schematic representation of the syntax and the explanation. And below is just one example of such pattern. So this pattern basically cuts out two different primers from read one. Then it trims the adapter sequences from the read two, and then extracts unique molecular identifier of length 19 nucleotides also from the second read. And actually, for all of the presets, for most of the presets, these tag patterns are already built in, so we already know the most suitable patterns for commercially available solutions. But for your own custom library, you can specify almost arbitrary, almost the pattern of almost any complexity. So MiXCR will handle this well. And after these sequences are extracted, UMIs and cell barcodes and so on, MiXCR runs refine tags and sort command, which corrects sequencing and PCR errors inside the barcode sequencing. This is extremely important because it diminishes the artificial diversity which is caused by these errors in barcodes, and this is extremely important. After this step was performed, the next step in the pipeline is MiXCR assemble, and this command basically groups all of the alignments by a specific gene feature, and this gene feature can be predefined one, such as VDJ region, which is the full length of the variable region of the receptor, or CDR3. So the clonotypes will be defined just by the CDR3 sequence, or it can be actually any arbitrary gene feature, such in this example. For example, here, this is a discontinuous gene feature with a gap in the middle. So this might be very handy in case your sequencing doesn't cover the full length of the receptor, but you still want to utilize the maximum information which you have from your sequencing. So this might be quite handy. And at the end, a Lineages file is formed, which contains all of the information about the clonotypes. And from this file, MiXCR export clones command can extract the information into a human readable file in tabular format, and actually one can specify all of the fields which are necessary. And here I picked several examples for export clones command for BCR repertoire analysis. And let me go through one of the examples. For example, this one, mutations detailed relative. And this field basically exports the nucleotide mutations in MiXCR notation with the corresponding amino acid mutations for each of the nucleotide mutation for CDR1 region in coordinates relative to V region. So starting from the beginning of the coding region of the receptor. And this is very handy, and this can actually save a lot of time on parsing and realigning and recalculation. So you use these fields because these are extremely useful and time saving. So with this, all of the upstream processing has been accomplished by MiXCR Analyze, and we already have our clonotype tables. However, before we go for downstream analysis, it's a good idea to make a little QC on our data set. And to do so, there is a number of built-in functions in MiXCR, first of which is MiXCR export QC align. And this command takes multiple CLNS files and generates this beautiful plot, which basically shows us how many of our reads has been aligned to a VDJ&C segment reference library. So for our libraries today, we see quite a nice percent, so more than 90% of the reads aligned to the reference library, which is the results of a very well optimized protocol in the VAT lab. So we can proceed with the next step of QC, which in this case will be the check for depth of coverage for each of the samples. And to do so, we can again use the same MiXCR export QC align command, however, with a flag absolute values. And this will generate the same plot, however, in the absolute number of reads. And for this case, we see that we have a little bit uneven distribution of coverage between our samples. However, for each of the sample, we have at least 1 million sequencing reads, which is far more than enough for our today's application. And to investigate this further, we can generate a different plot. Basically, this plot, which is generated with MiXCR export QC text command, also takes as input several CLNS files can take. And this plot generates the distribution of reads per UMI group. So in this plot, we can see how many of the UMI groups are covered by 1 read, by 2 read, by 3 reads, and so on. And it's usually a bimodal distribution with the lower peak attributing to the erroneous sequences covered just by several reads for one UMI group. And MiXCR automatically calculates the threshold for filtering out all of this dubious sequencing, which are probably just thresh generated in PCR or sequencing errors. And it works only with good quality cDNA molecules, which are covered by this number of reads per group. And as we see, all of our quality checks show that this data is very high quality, so we can proceed for the downstream analysis now. And first of the downstream analysis functions, which I'd like to talk today about, is the find alleles command, which infers the allelic variants for V and J genes using a powerful algorithm. And this step is extremely important for BCL receptor repertoire analysis because we have to definitely understand which of our mutations are allelic variants and which are the somatic hypermutations, because it will influence all of our downstream analysis and probably the implications which we have from our data. So this command does this crucial job, and it extracts, it actually can extract the donor-specific V and J gene allelic database to a separate file, to a FASTA file or a file in tabular format, but also to a file in JSON format, which will be compatible with MiXCR analysis, so you can rerun on this individualized library the analysis again in case you have different samples for this donor in future, for example. And it also realigns all of the clonotype sequences which were supplied to this command to this newly generated individualized V and J gene allelic database. So each clonotype from our original CLNS files will get the new assignment from the allelic database. And I also would like to emphasize that it can take several CLNS files as an input, but it's important to put here only the files relating to the same donor or to the same genetically identical model organism, for example, inbred mouse strain, and outputs the same number of CLNS files. And after we did this, we are ready for assembly and reconstruction of the immunoglobulin somatic hypermutation trees. And MiXCR does so with a powerful algorithm which utilizes all of the information which MiXCR generated during the previous steps of the analysis. So MiXCR knows all of the alignments and all of the mutations to the reference, to the accurately assembled reference database for each individual for a particular donor. So it distinguishes the allelic variants and somatic hypermutations. And most importantly, it knows the boundaries of specific regions within the variable receptors. For example, the boundaries of NDN region. So it knows where the added nucleotides are and how many nucleotides were trimmed from each of the V, D, and J segments. So it utilizes the information to group the clonotypes and build the lineage trees from the bottom up. And it also infers the intermediate sequences and most recent common ancestor and germline sequence for each of the trees detected in the data. Again, it can take several CLNS files, so it's easy for us, such as in this particular example, to use it for longitudinal data samples. And it outputs a SHMT file, which is a binary format file, again, which is extremely well compressed and computationally efficient. And from this data, we can export all of the information about the lineages found in our samples. And we can do this in several formats, first of which in UIC is in UIC format. This is a specific format for phylogenetic trees analysis. We can do this with export SHM trees UIC command and supply an output folder where a bunch of files in UIC format will be exported. And these files can be then easily be read into software which deals with this format, for example, GG3 package in R, or any tools which deal with the phylogenetic trees. And another variant here, another option is here, a tabular format input with the MiXCR export SHM trees with nodes command, in which you can specify the fields, or you can specify the fields within the preset file. And this works pretty close to export clones command, which is familiar with the earlier versions of MiXCR, and we discussed it today. And it basically exports all of the information about specific clonotypes, but with one distinction. It also outputs the information about the intermediate nodes. So intermediate nodes within the lineages also have most of these features which the clonotypes have. However, some of the fields are specific to the nodes and to the lineages. For example, here the N feature field exports the nucleotide sequence of this feature, and it could be also exported for clonotypes in export clones. However, in lineages, we have several different options. We can export the nucleotide sequence for the parent of this clonotype, which is the closest upstream node, for germline, which is the germline node, and for the most recent common ancestor. And we can also export the mutations from relative from parent or from germline or from MRCA, and distance from parent or germline or most recent common ancestor. And all of these things make it extremely handy and save a lot of time. For example, for the analysis for this webinar, I guess it saved me about several weeks or even months for parsing and realignment and so on. So this is extremely easy, and if you use this, check that the field that you want may be already present in MiXCR export, so you don't have to recalculate anything. So that is very handy for the downstream analysis. And before we go on with the lineage trees analysis, I would like also to mention that MiXCR now includes the post-analysis module, which actually can calculate the repertoire-wide metrics, and actually a number of different repertoire-wide repertoire-level metrics using the metadata supplied, and also accounting for differences in depth of coverage between the samples using a particular data type. So it can downsample by clonotypes, by the unique molecular identifiers, or even by cells for single cell applications. And here is one example of how it can be utilized. Here is the plot for hydrophobicity of CDR3 and the comparison between different states of our donor. In today's example, we see that there is some difference. However, it appears to be non-significant by the p-value, which was also automatically calculated by MiXCR. And so this is not the only one metric which can be calculated using post-analysis module. Here is the full list, and also for diversity metrics. But this is not it, because besides this, there is also a capability to generate V gene and J gene usage plots. For example, this beautiful heat map was generated using this function. And we can see, by the way, here that in two of the samples, one of the regions, in particular IGHV 333, is enriched. And if we take a closer look, we could see that these samples are coming exactly from the second COVID-19 time point, so the peak of response for the second COVID-19 infection. So this looks interesting, and let's remember this V gene. Probably we'll see it in our downstream analysis. And so I would like to continue with more in-depth lineage trees analysis, and start with the analysis of the abundance of each of the lineages which we detected in our data. And in these plots, each point represents just one lineage tree, and the values on these plots are basically the number of cDNA molecules in this lineage in this time point, in this replicate, and it's normalized by the total number of cDNA molecules in this repertoire. And when we compare the replicates from the pre-COVID-19, this is for second COVID-19 time point, we could see that even the lineages which are the most abundant in terms of number of cDNA molecules are not replicating well between the samples. And this is probably due to the fact that these lineages are represented by several antibody secreting cells which have high mRNA expression. However, they are not quite big in terms of number of cells which represent this lineage at this time point. So it's quite hard to sample them twice using this size of sampling which we used. So it doesn't replicate between two individual takes of blood at the same moment. However, at the peak of response replicate, and this is the central plot here, we see that the highly expressing lineages with lots of cDNA in them, they, on the contrary, do replicate between the samples, and this is due to the fact that at the peak of response, the active clonal expansions happen. So each of the lineages with the antibody secreting cells within starts to be presented not only just by several cells, but by a bunch of cells, and so it's much easier to sample them twice using independent replicates. And so that's why we see this increased reproducibility at the peak of response time point. And if we compare the pre-COVID-19 and peak of response replicates, we see that some of these newly emerging lineages with high abundance of IgH cDNA were also present, however, not many of them at the pre-COVID-19 time point. Actually, utilizing this information about sampling and the abundances, we could label those lineages which appear to be actively responding. So to label these lineages, I compared the variance between the replicates at the peak of response time point and the variance between the pre-COVID and peak of response repertoires. And also accounted for the number of lineages at the peak of response time point. And if we take these lineages and we track them back to other time points, we can see that some of these lineages which responded to the second COVID-19 time point, which is time point 7, also responded to the first COVID-19 infection at time point 4. So it's quite likely that we see here the recall response. So each of these lineages, so each line is basically one lineage. These lineages were also ones of responding at the first COVID-19 time point. So let's take a deeper look at some of these lineages, and let's start with the biggest one among all of the lineages in this data set. And here it is, and it utilizes IGHV333 gene, as we noticed on the V gene usage heat map, but probably it might be influencing this enrichment on this V gene usage plot, because it's quite a huge lineage. It has about 300 clonotypes in it, and a lot of total cDNA within this lineage. And it utilizes also quite long CDR3, and you can see here with the amino acid sequence alignments here, and this plot about frequency of mutations. Most of the mutations are concentrated in CDR1, CDR2, and also in the middle of framework 3 regions. And this lineage is a new one, which is specific to the second COVID-19 time point, to the second response to the COVID-19. And what's interesting, it decreases its abundance rapidly. Just in 5 days, the abundance in number of cDNA molecules representing this lineage is decreased by almost half an order of magnitude. And also, this lineage was represented by two different subisotypes of IgG, IgG1 and IgG3, and also with a little proportion of A1 clonotypes within this lineage. The next lineage is the second biggest one, and it's quite interesting because it's represented by two major sublineages, one which is a little bit skewed, and this may be a sign of a positive selection in here, and the other sublineage is rather flat, so it might be some local optimum here. And this CDR3 is also quite a long one. And again, what we see that this lineage is utilizing the same V gene, V333. So again, maybe these two lineages had major impact on this enrichment of this gene on our heat map, which we discussed earlier. And again, most of the mutations are concentrated in complementarity determining regions, with a little bit in framework 3 and some in CDR3. And in contrast to the previous one, this lineage was also present during the first COVID-19 infection. So in this response, although it wasn't the major one, it still responded. It increased its abundance, but genuinely, it increased its numbers on the second COVID-19 time point. And what's interesting, in the first COVID-19 infection time point, it was represented by A2 isotype, which is actually the last isotype in the human locus of IgH. So in these later time points, it should have been a separate and independent isotype switching event. So these cells are coming from some undetected memory cells in time point 5 and time point 6, because this switching couldn't happen from cells which we saw at this time point. And finally, I would like to show you a little bit smaller lineage utilizing different region, but it was one of the responders during the first COVID-19 infection. It's a little bit more evenly hypermutated through its length, and at the first COVID-19 time point, it increased its abundance dramatically, much more rapidly than in second time point. However, what's interesting, this is abundance in terms of number of cDNA molecules. And again, we see that the isotype is different at the later time points, and in the next COVID-19 infection, we see predominantly IgG1 and just a little bit of A1 here. And what's interesting, as I mentioned, we see a huge one huge member of this lineage at that time point 4, which is first COVID-19 time point. And on these alternative representations, all of the information for which I exported from MiXCR with just a little bit processing, we can easily see that this particular guy at time point 4 was just represented by singular sequence of A1 isotype, which had a lot of cDNA molecules in it. So it was just a big clonotype. And in time point 7 and time point 8, we see different members which apparently continue to hypermutate. And as we can judge from the number of non-synonymous somatic hypermutations here, it still acquires mutations which change it amino acid sequence, and actually quite a lot. And by this, we can assume that the affinity maturation is still ongoing in this lineage during the second COVID-19 infection. So this is it for today's talk, and I hope that I gave you a nice view of the new streamlined analysis pipeline with MiXCR, which can get you from the raw data to biological insights in just no time. And I hope that you appreciated this new support for barcoded data, either for unique molecular identifiers, cell barcodes, or even sample barcodes, and new algorithms for allelic variance inference and phylogenetic tree construction. I also hope that you like this short story about the detecting of the response to the viral antigens just through the replicates of PBMC with barcoded libraries, and we could just identify the responding lineages, just accounting for the cell sampling noise. and track them through the whole course of our study. So thank you for your attention, and I will be really glad to discuss these data or any other questions related to MiXCR and B-cell receptor analysis. So don't hesitate to use the Q&A button or the raise hand button, and let's keep in touch.
Okay, thank you very much, Artyom. Thank you for this very interesting talk. Can you show the next slide? Because I just wanted to no, well, that's the second slide of, yeah, this slide, because I just wanted to mention that you can ask any of your questions right in the Q&A section, and we already have a couple of questions which we're going to answer. Meanwhile, if you want to ask a question in person, just push the button to raise your hand, and once we will finish with the answers, answering the questions which you submitted through Q&A, we will unmute you, and well, yeah, you'll be able to ask. So we have a couple of questions, and I'll group those questions, sort those questions based on the timeline of the presentation. And well, the first question is how deep should be the blood sampling to obtain meaningful IG repertoires? Please, Professor Chudakov, it will be best if you answer.
Oh yeah, I'll do my best. So it's a sort of tricky question because, okay, what do you mean by meaningful? Depends on your aims actually. But altogether, in the full-length BCR profiling, we have been always like limited by the available depth of sequencing for the long reads, because for this full length we always had to use MiXCR, which is like 300 plus 300, or in case of our kit, it could be shorter. But still, you want to both capture some sufficient amount of molecules, and then you need to achieve coverage because you want to eliminate sequencing and PCR errors to track the true hypermutation history. So that's the basics. But from this point, it really changed now because we have the Illumina NextSeq 2000, which allows you to go much deeper. So we're not that limited in this respect now. We can go deeper in terms of number of molecules. But initially, it all starts from the cell sampling, actually. As you could see from Artyom's presentation. Artyom, could you jump the slide with the stochastics of sampling? So yeah, so the first, on the left, you can see these are basically two replicates of the blood taken in the same day. At the same time, there are large lineages which are not reproducibly captured. So what's wrong? How could it be? The point is that the cell sampling essentially in the B cell world, where like stochastics of sampling several plasma cells with lots of molecules inside, can really make it introduce quite a dramatic noise. That is why you need replicates to fill it. And that is why you should try to get more cells at start. The more cells you get, the lower is the noise, the more accurate you could identify the real signal in this world. And with sufficient coverage, you can do it now. So that's the game of several parameters, but altogether today we are much better here, both in respect to the kits and the software analysis and actually the sequencing machine. So we are just on the next level and can do it all together much better.
Mm-hmm. Well, thank you very much, Dmitry. Well, the next question is one second. It's 1 second. Yeah, it's a short question about in the presentation, had the PBMC been previously sorted?
Okay. For most of the samples here, we have not sorted any subpopulations from it. It was just bare PBMCs, and where there's just PBMCs from this PBMC's RNA was extracted. But for time point 4, we got additional samples of antibody secreting cells and memory B cells, which were, however, not utilized during this analysis.
Okay, thank you. Well, now let's just answer questions one by one because I can't sort them anymore. Too many questions. Okay.
Stan, maybe we should listen to raised hand question because
Well,
there's
I
one
want
raised hand.
I want first to answer all the questions we
Okay.
during the talk because they were submitted, and then all the raised hands will be answered. So the next question is: are those CDR3 metrics generated by MiXCR, for example, hydrophobicity? Yeah. Okay.
Yeah. All of this has been generated and calculated using MiXCR Postanalysis individual command. All of these metrics are calculated by MiXCR directly, and you can export them from the Postanalysis JSON file, which was generated during the Postanalysis individual step, and here it is as an output. And then plot this into a PDF file or SVG or PNG, supports multiple formats. So
Yeah.
yes, this was.
I just wanted to add that this hydrophobicity and, for example, strengths of interaction between CDR3 and antigen, that's the metrics calculated by MiXCR. That's an estimation based on just the amino acid sequences of the CDR3 region. So that's estimated hydrophobicity, estimated strengths, etc. And they're quite widely adopted in the literature, and we have links on the documentation to the original publications where they were discovered. Okay. The next question, what could be a reason for 100% load of all CPUs available, followed by overheating and PC hibernation after running the analyze command? Well, that's a good question, in fact, and that's a non-trivial question, because how MiXCR works, it utilizes all hardware available on the machine which runs MiXCR. So if you have 100 CPUs, then it will run everything in parallel on 100 CPUs. You can restrict this and put, for example, there is an option, and you can restrict the number of CPUs used by MiXCR, and then that will prevent the computer from overheating if there is not sufficient fan power inside. But basically, the reason is just because the data set is quite huge and processing takes time. It utilizes all the hardware. So once you have a laptop, it will overheat, obviously, or especially if you run like 1 terabyte of data on your laptop. Yeah. So it's best to run like analyze or whatever MiXCR commands for large data sets on large machines. Okay. The next question is, thank you for the great talk. Do you know if first and second infection was initiated by different COVID strains?
Yeah, we know this for sure. And first infection was initiated by Alpha strain, and the next one was by Omicron strain. So yes, this could be actually the lineages responding just for different strains. And maybe if it was the same strain, we could see that different picture.
Sorry, the question from me, how do you know that these are different strains?
Well, first, just by timing, and then for this donor, we have measurements at the second time point, at least. And obviously, Omicron wasn't present during the time of the first infection.
Okay. Okay. Yeah, and well, the Victoria who asked this question raised hand. So, Victoria, you're allowed to talk.
Thank you so much. I used to work in HIV field where we use different strains for immunization, different strains of virus. And it just made sense to me, the data that you got, that you have still maturation of the same lineages, right? That you had originally elicited by the first strain. Did I understand correctly? And then you had some new coming up?
Yeah, actually, we could see that some of the lineages can be tracked to the, just a second, can be tracked to the first time point. So yes, maybe some of these lineages which appeared during the first infection are maturating to the second one. However, talking about HIV, in HIV there is a little bit different story, because this lineage, which infects the patient, basically competes within this patient with the generated antibodies. Maybe you have seen the paper by Nur Mohammed, which actually investigates this question in depth, how these two evolutionary process compete with each other.
I'm thinking more from the immunogen perspective, not the actual strain of HIV, but the immunogens we used. So that would be independent, you know, kind of like a differently mutated protein that we added into the experimental animals. And then we would trace those lineages that would appear, you know, during the immune response in the animals, which would be similar, I guess, to this situation, right? When you have one virus and then the other virus. And so we saw some, you know, similar things where, you know, some lineages just get initiated de novo after the boost, or, you know, some lineages get more mature because of the boost.
Both situations can be present. So as we can see in this example, some of the lineages which had increased in abundance at the first COVID-19 time points didn't respond at the second one, and just new ones appeared. However, some of the lineages participated in the recall response.
Okay, thank you. Cool data. Thank you.
Thank you for the question. So the next question, Wen Zhao, you raised your hand and I'm going to unmute you. So yeah, I have unmuted. Yeah, please.
Oh, can you hear me?
Yes.
Oh, actually, I put in the chat window. I want to ask you, do you guys find a allele program first before you run SHM analysis, or you can actually skip this find allele step? The second question is, when you do this SHM lineage analysis, do you remove this new allele, nucleotide changes, so it will not be counted towards your SHM or lineage analysis?
Oh, thank you. These are really the good questions. And thank you for this chance to elaborate on this. Yes, first, we should definitely run find alleles command before we run the immunoglobulin trees inference commands, find SHM trees. This is exactly for this reason, to account for the allelic mutations, not for them not to be included as somatic hypermutations in the lineage reconstruction step. And actually MiXCR has a built-in check that you have run the find alleles command, because if you don't do this, if you don't have the proper reference and all of the allelic variants inferred, you will have basically the wrong lineage trees, because these mutations, they greatly impact the process of the reconstruction of lineage trees. If you have the allelic mutations present, the trees will be incorrect, and also several metrics. For example, if you build some phylogenetic analysis, such as site frequency spectra, again, allelic variants will make these plots and these kind of analysis just wrong. So yeah, it accounts for allelic variants, and it distinguishes between somatic hypermutations and allelic variants for lineage trees reconstruction.
Okay, thank you.
Thank you very much for this question. So now I'm going to continue with the questions submitted to Q&A, and the next question is, has MiXCR right now have a pipeline to analyze 10x genomic data with antigen barcode? Well, first, yes, we have a pipeline for the analysis of 10x genomics VDJ data, and we also have a pipeline for analysis of 10x genomics 5' gene expression data, but not the gene expression analysis, but just extracting of repertoires from gene expression data. So I guess yes, the answer is yes. And there is a one-line comment which you can use, just MiXCR analyze 10x VDJ, and then that's it. Okay. The next question Thank you. Sort of the basic pipeline question, should input reads be quality trimmed? Let me answer. No, you don't need to do any pre-processing. You just need the raw sequencing file, just right from your sequencing machine. You can even perform the demultiplexing with MiXCR, but well, if you already have a demultiplex FASTQ file, that's fine, and nothing else. Just the raw sequencing data, and then MiXCR will do everything for you. Okay. The next question, we have data with sequenced receptors available soon, but so far we have single cell data with no receptor sequencing, so we use Trust4. What Trust4 output would we use as input for B-cell analysis? Well, MiXCR can't take output from Trust4 or, well, any other program. It takes input, it takes just raw sequencing files. So if you have raw sequencing files, then it's fine. MiXCR will take raw FASTQ files and do everything for you. So you don't need to do any kind of pre-processing, whether with Trust or with any other software. And in case if you have RNA-seq data or single cell data, it's still possible. So you can run MiXCR with RNA-seq data, shotgun DNA data, whatever, and it will do the same. And there are ready-to-use presets for any kind of protocol and data type. Next question. For the lineage trees, are the inner nodes and root node inferred purely computationally? Yeah, they are synthetic. What if both the most common ancestor itself and the expanded clones are found in the sample? So
let me take this one. Yes, actually, we have an example when their intermediate nodes, the inner nodes, are present in the data and MiXCR utilizes them. Yes, that could be possible, and they will be present in the lineage trees as just as the synthetic inferred computationally. So yes, and even in this data set, we have such an example, several examples of this. However, this is not the very common situation. However, for some of the most interesting trees, this could happen, and it happened with several trees which respond to these COVID-19 challenges as well. So basically, the answer to this that the inner nodes can be found in our lineages, and MiXCR handles them like as other inferred computationally ones.
Thank you for the answer and for the question. The next question is it possible to enter this workflow midway? For example, provide BOM file instead of FASTQ, or clone sequence and count tables for the SHM analysis? Well, you can provide BOM input instead of FASTQ, and MiXCR will start from the BOM file, but what MiXCR will perform, it will extract raw sequences from the BOM and then do the whole pipeline using the raw sequences. So that's no difference whether you start from the BOM or from the FASTQ. And regarding the second part of the question, clone sequence and count tables for the SHM analysis. Well, basically, the answer is not, because MiXCR treats clonotype. The information about clonotypes used inside MiXCR is much more, well, rigorous than just the sequences of clonotypes and their counts. It stores all the alignments to the reference, allelic variants, and, well, tremendous amount of information which is used to build reliable trees, reliable allelic variants, etc. But what you can do as a workaround, if you don't have raw sequencing data, you can put those clones as like in a FASTA file and submit to the whole MiXCR analysis pipeline as like a raw sequencing data. So that's possible.
I had such experience. However, I really recommend you to find the initial data set if you can, because MiXCR utilizes the quality information. It handles the UMIs more carefully. It utilizes it to assemble clonotypes. So in case you have the raw data, the quality will be better, basically. And it utilizes all of this information for building trees and allelic variants. So.
Okay, thank you for the question and for the answer. the next question, is there a way to get HLA typing information with this analysis?
Well, I guess this is a bit a question related to T-cell receptor repertoire world. So maybe Dmitry would take this
I
one.
guess it's a more direct question. So currently there is no functionality for HLA typing in MiXCR. We keep it in mind, but we are focusing on other things right now. So.
Yeah. Okay. Thank you for the question and for the answer. Next question. What is the distance and method for UMI and barcode collapsing, and can this be modified by the user? Well, let me answer this question. Well, this is a very good question and a very deep question, because the algorithm for UMI collapsing is non-trivial. Well, basically, MiXCR uses alignments, and what MiXCR does first is alignment and extraction of barcode sequences. So the collapsing of UMIs occurs at the clonotype assembly step. That's what we call pre-assembling. MiXCR uses alignments, and it uses all the alignments from the same with the same UMI to build a consensus alignment or multiple consensus alignments if we have a low diversity of UMIs. And that's done on the level of alignments. Once you know alignments to the reference with all indels, whatever, that's much more reliable compared to pre-processing when you operate with the reads. And the algorithm used, well, I can't explain this algorithm in a few sentences, unfortunately, but we can follow up because it's complicated. And there are plenty of parameters which you can tweak, tune, and play with this algorithm to achieve what you're going to achieve, what you want to achieve. But basically, we already optimized it for most of the protocols, data types with many samples. So the parameters which are there by default are just, well, the optimal ones. Next question, and that's going to be to the last question. If we are running synthetic calibration standards with the samples, is there a way to compare the data or do some kind of downstream analysis?
Well, okay. Basically, if you know the sequence of your particular calibration standards, they will be in the output files from MiXCR, both in CLNS and the human readable formats. So after you export the tables, you can find your calibration standards and utilize whatever software you use for the downstream analysis, such as R or Python or anything. So basically knowing the sequence is the key, and MiXCR will for sure find the calibration standard within samples.
Okay, thank you for the question and for the answer. The next question is, I can see where the clonal trees are helpful to compare the same patient over time and response. What are the best B cell receptor comparisons when there are different patients samples, for example, different atherosclerosis samples? Dmitry, I believe that's better to answer for you.
Yeah, let me try to guess. In contrast to the viral infection of vaccination with a strong vaccine, when the amplitude of response is high, and you can use the clear, more or less, time points for comparison and extraction, the responding lineages, in some sort of diseases like autoimmunity or autoimmune-like, where the expected amplitude of response is very small, it's a tricky thing. So, and if it is a different patient sample, so you're going to extract information from like huge amount of donors and try to find there the characteristics, antibody motifs. It's the next level of complexity. There are ways to do it, but I would say it's a matter of separate investigation and separate algorithms for the post-analysis to find the motifs across million sequences and thousands of patients. So different story.
Yeah,
but
and
you could technically you could obtain all the best repertoires and lineages with this arsenal of tools. but the question you're asking refers to further investigations. So it's on the basis for this.
So it will be a completely different thing. It will be just clonotype clustering, not the lineage trees. So from a totally different perspective.
Okay, thank you for the question and for the answer. And this is the last question is with MiXCR4, I understand there is no need for MiGEP. Is UMI analysis more accurate in MiXCR? The answer is very simple, and the answer is yes. It's much more accurate, much more powerful, much more flexible, provides you much better reporting, quality controls, etc. Our old MiGEP tool, well, is deprecated, in fact. So we don't recommend it to use, and we recommend to switch to MiXCR4 because it's just much better and more reliable in plenty of ways. Yeah. Okay, thank you very much. That's all questions we have. We don't have any more raised hands for now. So please let me just remind that if you have any questions related to our today webinar, to our topic, or to MiXCR platform, you can always reach us using our support email, or you can text me directly on my personal email, which you received with the invitation to the webinar. And you can check the recent documentation about MiXCR on our documentation portal, docs@mylaboratories.com. We will send every attendee all the materials, including the link to the video recording, slides, and scripts used by Artem for the downstream analysis of the trees. Thank you, Artem. Thank you very much. And thank you, everyone, for joining our today webinar. We'll keep in touch. Thank you.
Thank you, people.
Thank you.
Goodbye.
See you.